Hello everyone, welcome to my channel Lifescience with Nimedita. So in this video, I'll be solving and discussing previous year KSET Lifescience question paper. So without any further delay, let's get started. So we just move to the first question of KSET Lifescience question paper. So here, you can see the first question here. An open reading frame contains 333 nucleotides. The number of amino acids in the protein synthesized from this ORF will be. So here, ORF is nothing but what? That is open reading frame. Okay, so this question is related to molecular biology. That is translation process. During the translation process, mRNA is going to synthesize protein, right? So during the protein synthesis, amino acids are there, right? That is 3 codons are codon for a single amino acid. So based on that, what happened? Proteins will be synthesized. Okay. So here in this question, they are asking 333 nucleotides are there in the open reading frame. Okay. So they are asking what? The number of amino acids in the protein synthesis from this open reading frame will be what? Okay. So usually 3 codons are there, right? So for example, AUG is there. So this AUG we call as a start codon, right? So this start codon is always codes with which amino acid, that is methionine, right? So like this, several codons are there which will code for a specific amino acids. Okay. So here what they are asking, 333 nucleotides are there. So these 333 nucleotides are coded with help of 3 codons. Okay. So 333 nucleotides are there divided by 3. So this 3 represents what? This 3 represents 3 codons. Okay. So here 1, 1, 1. Okay. So don't rush. Here already 111 option is there. But this 111 is not a correct answer. Why? Because in open reading frame, start codon is there as well as stop codon is there. Right? So here, in this 333 nucleotides, remaining 3 is there, right? The last 3 codons, that is last 3 nucleotides are coded with the stop codons, that is UGA, UAA, and also UAG. Okay, so any of the stop codons are there in this 333 nucleotides. So that's why 333 minus 3. So this 3 is what? That is stop codons. So here 330 divided by 3. Because we have to leave the stop codon, because these stop codons does not code with any amino acid. Okay. So 3 ones are 0. Okay. So here we just, if you are in rush for this question, means what we will do? Divided by 3, 1, 1, 1. Okay, already 1, 1 is there in option. We just stick option A. That is wrong, because last 3 nucleotides are coded with the stop codons. So that's why answer for this question is option B, that is 110. Okay, so like this you have to solve this type of questions. Okay, and also in translation itself, there are many codons are there. They can be asked a specific codon codes for which amino acid, like valine code for like a valine code for, name the codons for the valine, like that also they can ask. Okay, so like that, like this question will be raised for a particular questions related to molecular biology topic. Okay, so next move on to the next question. Okay. So the next question, plant movement that takes place in response to touch stimulus is called as what? Anyhow, response to the stimulus. That movement is called as what? Growth of a plant which is response to a touch. So that is response to a contact. So that type of movement we call as what? That is thigmotropic movement. Okay, so thigmotropic, there are 2 types of touches there, that is positive touch and also negative touch. So the positive touch means what? That is towards the touch. Okay, and the negative touch is nothing but that is away from the touch. Okay, so similar like this, some other options are there. Chemotropic movement means which responds to the chemicals. For example, pollen growth development. Okay, so during the pollen tube development, the pollen tube is there, no? Okay, so here the pollen tube towards the ovule, the growth takes place. Why? Because where the food is rich, the area where the food availability is rich, in that moment where the chemicals are rich, in that moment what happens? The movement takes place. So that type of movement is called as chemotropic movement. Example, the pollen tube generation or formation. Okay, so next is the hydrotropic movement. Hydrotropic movement is nothing but what? That is towards the water. Example, roots. Okay, so next, the osmotropic movement. Osmotropic means the movement of dissolved organic components for nutrition. Okay, so that is example for osmotropic movement. So the answer for this question is what? That is thigmotropic movement. Okay, so next, move on to the next question. Yeah, there will be one question related to this type, that is genetics. It is as you dominant like that. Okay, so color blindness is a X-linked recessive disorder. If a child is born to a normal father and a carrier mother, what will be the probability that the child is color blind? First, read question carefully. Okay, so this is a X-linked recessive disorder. X-linked recessive means anyhow female having XX chromosome, males having XY chromosome. So X-linked recessive means if X is a carrier, for example, male contains XY chromosomes. If X is a carrier, the male child will be affected. Okay, so just write down here. Okay, so what there is saying, colorblind if a child is born to a normal father. So here father is normal, that is XY, normal father, and a carrier mother. So mother having two X chromosome, right? So in this two X chromosome, one X chromosome is carrier. Just write with another color. Okay. So here in XX chromosome, one X chromosome is a carrier. Okay. So after one generation, what will be the probability that the child is having color blind? Okay, just do the calculation here. X. So one child, that is a normal daughter. Next, X. Next, carrier. So next is a carrier daughter. Y. X, that is XY. Normal son. So next, Y. That is X carrier and also Y. That is color blind son. Color blind son. Okay, so in this question, what they are asking, what will be the probability that the child is a color blind? So what is the probability? So here 4 is there. So in this 4, only one child is affected. That is 1 by 4. So if people say here also the carrier is there, but this is a female child. So even though one chromosome having a carrier gene, it is not expressed. Okay, so only in the males it is expressed. Why? Because males are having only one X chromosomes, females are having two X chromosomes. Okay, so what is the answer for this question? That is 1 by 4. Okay, so in the 4 children, one child will be affected. You can see colorblind son. So for this type of questions, you have to solve like this. Okay, so next question. Yeah. Okay. Which techniques are used to study the transcription process in the cell? So transcription process. So just write on the central dogma of molecular biology. DNA to RNA, RNA to protein. Okay. So the process of conversion of DNA to RNA is called as what? That is transcription. And the process of the conversion of RNA to protein is called as what? That is translation. Or else protein synthesis. And the conversion of DNA to DNA is called as a DNA replication. And the conversion of RNA to DNA is called as what? That is reverse transcription. So this one we call as a central dogma of molecular biology. So in this question, what they are asking, which techniques are used to study the transcription process? So transcription means which process? Transcription means which process? That is RNA synthesizing process. Okay, so northern and western blotting. So here, northern blotting is always used for the RNA and western blotting for protein. But here only they are asking transcription. Okay, so PCR and restriction digestion. PCR means polymerase chain reaction. This is for DNA and also RNA multiplication. And restriction digestion means, that is restriction digestion means which is an enzyme which is used act as a molecular scissors. Okay, so next is the northern blotting and in situ hybridization. So northern blotting is for RNA and in situ hybridization. So this is also for the localization of a nucleic acid for both DNA as well as RNA and RNA. Okay, so this southern blotting is for DNA and ELISA test for AIDS. That is also for RNA. So in this question, only they are asking the transcription. Transcription means the RNA. So where only RNA appears, you can see northern blotting and also in situ hybridization. So here both DNA and RNA we can identify, but here they are asking only transcription. That's why here option C is a correct answer. So which will be helps to study both northern blotting, that is only RNA, that is northern blotting, and also in situ hybridization. Okay, so just move to the next question. So the fifth question, yeah. Radioactive sulfur was used to label the T4 phage coat protein when the phage was infected to equalize cells and the cells were precipitated by centrifugation. S35 labeled protein was found with what? So this question is related to Hershey and Chase experiment. So this is an experiment they conducted to prove DNA as a genetic material. Okay, so here in this experiment, they used bacteriophage. Okay, in this bacteria, they use two radio-labeled molecules, that is phosphorus 32 and also sulfur 35. Okay, so anyhow, when it comes to the DNA and also protein composition, DNA having the phosphate backbone. Okay, for the detection of DNA, they use phosphorus 32 radioactive labeled molecule and sulfur where it is present in the amino acids. Okay, in the proteins is what? It's a chains of amino acid, right? So cysteine, methionine are having the sulfur in their structure. So this sulfur 35 radioactive labeled molecule helps to indicate proteins. Okay, so here they took this bacteriophage, Hershey and Chase. So this bacteriophage, okay. So first one, they took this phosphorus 32 DNA and also they injected this to the cell. Okay, so here DNA is there, which will enter inside the cell. So after the centrifugation you can know, right? Okay, once the centrifugation is done, the pellet will be settled and supernatant will be present in the top. Okay, so here supernatant is there and also pellet is there. Okay, so here once this experiment is done, this phosphorus 32 is there, right, which will be present in the pellet. That is, this DNA is there, right? DNA enter inside the bacteria. Okay, with the help of many gene transfer methods are there, right? So because of that, this DNA enter inside the bacteria. So based on the density, DNA having the more density, that's why it will be settled it at the bottom. Okay. So next, they took one more bacteriophage. Okay. So they are adding sulfur 35 to this bacteriophage. Okay. So next again, which will be infected to a cell, bacteriophage. Okay. So this bacteriophage contains sulfur 35 protein coat. Bacteriophage contains protein coat, right? So in this protein coat, which contains a sulfur 35. So after the centrifugation, once the centrifugation is done, so we can able to identify the sulfur 35 in the supernatant. Okay, that is outside of this outside of the cell. Inside the cell, DNA is there. Outside the cell, protein is there. So inside the cell, only genetic material transformation takes place. So that's why it should be settled at the bottom. And here, this bacteriophage protein coat is there. Protein contains sulfur. So that's why, based on the density, which is present on the supernatant. Okay, so what they're asking the question is in the T4 phage coat protein. When the phage was infected to equalize cells and the cells were precipitated by centrifugation. So after the centrifugation, you can see S35 labeled protein was found in where this S35 labeled protein found, which is found in the cell supernatant. That's why option B is the correct answer. Okay, so this series will help you to boost your preparation. So like this, I just start question paper solving series daily. I'll solve five questions related to KSET. Okay. So if you found this video is helpful, just subscribe to my channel and let me know in the comment box if people need more video like this. Okay. So thank you so much for watching.